Today's the 26th. Okay, so you have constructed DNA, and we've got lots of fine, lovely pieces of DNA out there. Lovely pieces of DNA. What I would like you to do is compare the DNA that you and your partner created to the strand created by the other set of people at your table. If you are alone at a table, then go ahead and compare it to somebody at a different island. Yes, that is a good question. We're going to talk about that. That is a good question. So it would be unlikely that anybody would have absolutely identical DNA molecules. In most cases, we very often end up with ones that are similar, just sort of random chance. But we can have identical DNA molecules that would be like the DNA of twins, where the molecules top to bottom are identical. You could superimpose them. If you have questions, put them on the back channel. But most often we see some variation. Now, if we started to string all of these together, we might begin to approach something like the length of a short section of DNA. Remember, the DNA molecule is enormously long. Now, if we think back to bacteria, what was the process that they used to reproduce? It's asexual reproduction. It's called binary fission. So, good. When bacteria perform binary fission, what was that first step? They copy something. They copy their DNA. They copy their DNA, and that copying is called replication. Okay, that's called replication. What does the prefix re mean? Again. To replicate something is to copy it. So what we're going to do is replicate your DNA. But first I want to walk through the steps of how that happens. Now, to simplify the process of replication, I have drawn this molecule of DNA untwisted. Okay? It isn't actually untwisted, but I'm drawing it that way to simplify looking at replication. So here's our untwisted molecule of DNA, and we've got two halves. And the first thing that you have to do in order to copy this is you have to unzip. Okay? So there's an enzyme. An enzyme is a chemical that exists inside the cell that comes along and it travels just like a little zipper, and it literally opens up the DNA molecule. It's called DNA polymerase. It doesn't matter. Okay? But it opens up the DNA molecule. So now we have this. So what I'd like you to do is you and your partner take your DNA molecule and open it up. Unzip it. Unzip it. So here's the cool part. From this one piece of DNA, we have a model to make two new pieces of DNA. Do you know what always matches a G? A C. So every place we see a G, we can put a C. Do you know what always matches an A? A T. Do you know what always matches a C? A G. Do you know what always matches a T? A. I forgot a T. Okay, so guess what? We now, I'm going to change this to a different color. I'll make this sort of, no, that won't work. I'll make it grayish. Whoops.
If we match up with this one, no, I'm wrong. But we have the same pairs.
I now have one new piece. Hey! Thanks. I now have one new piece of DNA. And guess what? Out of the same exact thing, I get my second strand replicated. So one strand of DNA will generate, through replication, 2 new strands that are identical to the original strand. Now, I sort of color-coded that sugar backbone to be different, only so that we can see that what I get are 2 new identical strands. There's no difference in the sugar backbone on the new strand. But, have you figured out what the white pipe cleaner is for? That's going to be the sugar backbone of your new strand. So each partner should get a white sugar backbone. Hey, don't throw it. Don't throw your sugar backbone. And half of your DNA molecule. So now you're going to replicate your DNA using that white backbone. What you have just done is to replicate DNA. You started off with a single strand of DNA, and if you compare it to your partner, you should have 2 identical strands of DNA, 2 identical segments of DNA. Okay? The half that you just replicated is called the complement. So it is complementary, identical, able to be superimposed. So if we look at a DNA sequence, if we look at A, T, C, 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 T, A, G, whoops, G, we can find the using the what rule? What's the rule called? Base pairing rule. So complement to A is T. Complement to T is A. Complement to C is G, 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 A, T, C. Okay. That's DNA replication. That's pretty straightforward. Now, what I would like you to do, Danka, tuck your DNA strands away in a notebook. Because tomorrow we'll start talking a little bit about mutations. We'll start talking about things that can go wrong. So I'm going to want you to keep those because we're going to come back to them a couple times. Please leave the cups with the bases intact because the next class will need them. In the meantime, and you know you guys have a 2 hour delay tomorrow, right?
Yes.
It means nothing for us because you never miss a minute of this class all year long. You are tortured by me for a full 60 minutes.
Yes.
Yes. So what I'm going to do now is give you your vocab packet for this chunk. This is, remember, why are we talking about DNA and bacteria? We're talking about it because we want to know how DNA can actually become resistant to an antibiotic. So to do that, we have to know how they build their cells. And to understand that, we have to understand their genetic material. So that's the big question that we're coming back to. But to do that, we got to get through some detail stuff about the DNA. So this is sort of background on what they do. All right, I'll hand you the vocab packets. You have the rest of the time to work. Part A and B are due on Friday.